Can I please invite our last speaker to the stage for this session, Miss Ivy Mutai, who's been working with the University of Leicester and the University of Nottingham for her flash talk entitled Phage Biosanitizer to Reduce the Spread of Antimicrobial Resistant Salmonella Species on the Poultry Farm Equipment Surfaces. Thank you.
Thank you for the introduction. So I'm going to present on a phage biosanitizer to reduce the spread of antimicrobial resistant salmonella species on the poultry farm equipment. These are my team members. And this work I was doing at the University of Nottingham before I moved to Becky Meyer Center for phage research at the University of Leicester. Salmonella causes approximately 95 million illnesses annually, together with around 50,000 mortality rates. And thank you to the previous presenter who gave a very good introduction about antimicrobial resistance and bacteriophages. And bacteriophages have been recommended because they have no residues, and they can also be upscaled in industrial setting. So our aim of our study was to isolate and characterize these phages that could be used as biocide or biosanitizer in poultry farm equipment surfaces. Our experimental design. So we revived Salmonella strains that were from our inventory collection, and we collected poultry samples from environmental samples, and we also did isolation of phages through standard methods of phage isolation. And baseline is to determine which phages are the best through host range analysis against the 108 strains, and then we did whole genome sequencing of these phages to determine which ones of them are lytic, because we want to proceed with them. And we selected now candidate phages for the biocontrol study. So in total, we isolated 35 novel Salmonella phages, 29 were sequenced, and we proceeded with 2 of them. And these were based on the host range, as I've mentioned. And on the top, this other side, this was our experimental design. We had 2 types of poultry farm environmental contact surfaces, PVC and the stainless steel. And we had 2 concentration of bacteria, 9 logs. It was supposed to be 8 logs, that was an error, and 4 logs for the biofilms to form. So the family attached was for 48 hours, which was for 2 days. And the biofilms was, for this low bacterial concentration, was 4 logs, that was going for 7 days, with media replenishing at 1, 3, and 5 days, before we treated with phages for 24 hours. So these were to emulate the actual environmental conditions of poultry environment in the UK. So the temperature was 22 degrees, 22 plus or minus 1 degree. So basic characterization of the phages, the burst size of the 2 phages here, 20.9 plaque forming units per cell, and the other one had 123.7. So I had mentioned we used 1 commercial phage cocktail from PhageGuard to just give us a comparative overview of how our phages were working on the poultry contact surfaces. This was a genomic synthetity or sequence alignment to look at in comparison with the reference genomes. And these were our phage. This was one of our phage in this, and these were the reference sequencing, and we could see that they had a bit of variation in their tail region, showing us that they could be targeting different receptors on the surface of the bacteria, which is good to incorporate. 1 minute. This should have been the best slide to present, but yes, 1 minute. So on the effect of the low bacterial concentration for the cocktail, low bacterial concentration for the 7 days, we observed that the cocktail actually worked better as compared to the individual phages. And across the two surfaces, the high bacterial concentration and the low bacterial concentration, the cocktail performed the best. And one of the phages, if they were monophages, 1-PSJ, which performed the second best if it was a monococktail. And we also did physical chemical to determine if we are going to proceed to industrial production of these phages, how stable are they? And effect of temperature, the optimum was 37. And one of them went up to 65, which was really interesting to see a phage very active at 65 degrees. And the pH, none of them were active at 2, which was expected. And storage, over 1 month storage, we observed that -20 is not the best to store the phages. And the lyophilization, we wanted to look at long term, how do we store these phages? Are we seeing any reduction in titer once the phage has been lyophilized? And in real sense, it's a sustainable method to store the phages for long term. So this project received funding from Canada and BBRC, UK, and a few recommendations, Paul Barrow for giving us the collections of the salmonella, and the Phage Guard Company for donating the commercial PGS for the comparative study, and Professor Christopher from University of Nottingham for statistical analysis. Thank you very much.